This is the first of 3 chapters that will introduce you to the cardiovascular system. The cardiovascular system is an internal network that links all parts of the body. It includes the heart that acts as a pump to move blood, and blood vessels which provide a route for the blood. So we'll begin our discussion of the cardiovascular system with blood. In this lecture, we'll learn about the components and function of blood, as well as plasma and red blood cells, and how we can use characteristics of red blood cells to group people into blood types. We'll also discuss the structures and functions of white blood cells and platelets, and how mechanisms that control blood loss after an injury. The circulating fluid of the body is called blood, and it's a specialized connective tissue that contains cells suspended in a fluid matrix. Blood has 5 major functions. It transports dissolved gases like oxygen and carbon dioxide, nutrients from the digestive tract, hormones to target tissues, and metabolic waste to kidneys for excretion. Blood regulates pH with a series of buffers and regulates calcium and potassium ion makeup of interstitial fluids. Blood can restrict fluid loss at injury sites by vasoconstriction and clotting factors. It also defends against toxins and pathogens by transporting white blood cells, the main component of your immune system, and by transporting their products, antibodies. It stabilizes body temperature as well. Blood absorbs the heat from skeletal muscle contractions and redistributes it to other tissues. Whole blood is composed of formed elements, like cells, suspended in a fluid matrix called plasma. In general, blood is a little above body temperature at 38 degrees Celsius. And blood is thicker than water, about 5 times more viscous. Remember, viscosity refers to thickness or stickiness. And this is caused by plasma proteins or formed elements. Also, the pH of blood is slightly alkaline in a range of 7.35 to 7.45. Let's take a closer look at the fluid matrix of blood, plasma, which makes up about 55% of the volume of whole blood. Along with interstitial fluid, plasma makes up most of the extracellular fluid in the human body. 7% of the volume of plasma is plasma proteins, such as albumins, globulins, and fibrinogen. Albumins are probably the most abundant, and they're responsible for maintaining osmotic pressure of plasma. Globulins act as transport proteins and as antibodies. And fibrinogen functions in blood clotting. It can be converted to fibrin. About 90% of plasma proteins are synthesized by the liver. Now, plasma minus clotting proteins like fibrinogen is called serum. Plasma also contains hormones, nutrients, gases, and about 92% of plasma is water. We've reached a checkpoint. See if you can test your knowledge so far. Now let's talk about the most abundant formed element in blood, the red blood cell. Red blood cells make up the majority of the formed elements, about 99.9%. Red blood cells are typically measured in a red blood cell count in cells per microliter. Men have a few more than women at about 5.4 million per microliter, and women have about 4.8 million red blood cells in a microliter. When we refer to the amount of red blood cells, we typically refer to it in terms of a percentage of the whole blood, which is a hematocrit. Men have a hematocrit of about 46%, and women have a hematocrit of about 42%. Red blood cells have a very unique biconcave shape, which provides advantages. It increases the surface area and increases the rate of diffusion. And it has an increased flexibility, which gives it the capacity to squeeze through narrow capillaries. During red blood cell formation, organelles are typically lost. Therefore, red blood cells cannot go through a normal cell division. They can only rely on glucose from plasma for energy. Notice how there's no nucleus or mitochondria present. 95% of red blood cell intracellular proteins are a pigment molecule called hemoglobin. Hemoglobin transports oxygen and carbon dioxide. Hemoglobin is composed of two pairs of globular proteins called subunits that are labeled in this picture as alpha and beta chains. Each subunit contains a heme with an iron atom. Oxygen binds to heme. Carbon dioxide typically binds to the globular subunits. The oxygen-heme bond is a fairly weak bond. Under conditions of high plasma oxygen concentrations, the hemoglobin will gain in oxygen until it's saturated, as blood circulates through the lung capillaries. Under conditions of low plasma oxygen and high carbon dioxide, hemoglobin releases the oxygen, and this is how oxygen is delivered to cells throughout systemic capillaries. Red blood cells are subjected to severe physical stress, well, at least in comparison to an average cell. They're squeezed through small capillaries and bounced against walls of blood vessels, making a complete circuit from heart to peripheral tissue and back again about once a minute. And take into account that they don't have any repair mechanisms, which is why a typical red blood cell only lives about 120 days before it needs to be replaced. About 1% of all red blood cells are replaced each day, which, if you do the math, is about 3 million new red blood cells that enter circulation per second. If red blood cells hemolyze, or burst open into the bloodstream, the hemoglobin breaks down and the kidneys will filter it out and excrete it in urine as waste. However, most red blood cells are phagocytized in the liver, spleen, and bone marrow, and then the hemoglobin components are recycled. And that's what's being demonstrated in this figure. There's 3 steps to hemoglobin recycling. First, globular proteins are broken into amino acids. The heme is stripped off of the iron, converting hemoglobin to biliverdin. And biliverdin is then converted to bilirubin, which is released into the bloodstream. The liver absorbs the bilirubin and it becomes part of bile. Now, iron can be stored or released into blood to bind with transferrin, which is absorbed by bone marrow and used in new red blood cell formation. The process of red blood cell formation is known as erythropoiesis. In adults, erythropoiesis only occurs in the red bone marrow or the myeloid tissue, specifically in the vertebrae, sternum, ribs, scapula, pelvis, and the proximal limb bones. Red blood cells result from a division of hemocytoblasts, or multipotential stem cells, in red bone marrow. These multipotent stem cells give rise to myeloid stem cells, some of which proceed through a series of stages to mature erythrocytes. One such stage is an erythroblast, which are very immature red blood cells that are actively synthesizing hemoglobin. After this stage, the erythroblast sheds its nucleus and becomes a reticulocyte. At this point, they enter the bloodstream to complete their maturation into a red blood cell. This figure shows the big picture of blood cell formation. Remember I said that hemocytoblasts are multipotential stem cells, meaning that they can produce red blood cells, but that's not all they can do. They also have the potential to form platelets and white blood cells, so you'll see this slide again later. In order for this process to proceed normally, a steady supply of amino acids, iron, and B vitamins is required. Erythropoiesis is stimulated by low tissue oxygen called hypoxia. You remember which organ monitors for hypoxia? In the last chapter I told you it was the kidney. So anytime hemostasis is disturbed, like when blood flow to the kidneys decreases, or anemia occurs, or oxygen content of air declines, or when damage to respiratory membranes occurs, anytime hemopoxia is detected, The kidney will release the hormone erythropoietin, which is abbreviated EPO. Target tissue is the myeloid stem cell tissue, and that's going to stimulate an increase in cell division, which will speed up the rate of maturation of red blood cells. More red blood cells means more oxygen content in the blood until homeostasis is restored. So we've reached one more checkpoint. You could pause the recording and see what you've learned. The ABO and Rh blood typing system is based on antigen-antibody interactions. An antigen, or agglutinogen, as your lab manual refers to them, is any substance that can elicit an immune response, or specifically an agglutination reaction. Most often antigens are proteins. Now, all of your cells have antigens on their surface. Some of these antigens are A, B, or those proteins involved in the Rh group. And these are used to group people into blood types. Type A blood has antigen A only. Type B blood has antigen B only. Type AB blood has both the A antigens and B antigens on the surface of the red blood cells. Type O blood has neither the A antigens nor the B antigens. Rh positive is a notation that indicates the presence of any of the Rh antigens. Rh negative is the absence of any of those antigens. The other half of that reaction is the antibodies, or the agglutinins. Antibodies are small blood proteins that are generated by white blood cells as part of your immune system. At any one time, you have many different flavors of antibodies floating around in your blood. Because antibodies are very specific and will bind to only one antigen, so for all the different kinds of antigens out there, your immune system can, and maybe has already, made an antibody that will recognize and bind to it. But your immune system is smart. It has learned to tolerate the antigens on your own cells, and so it will only attack foreign antigens. This is the case of antigens that make up the blood type groups. Those people with type A blood have anti-B antibodies in their blood. People with type B blood have anti-A antibodies. People with type AB blood have neither antibodies in their blood, whereas type O blood has both antibodies in their blood. The presence of these antibodies is why, before a blood transfusion, the blood type of a donor and recipients are identified. This is one of the activities we do in lab. If an individual receives blood of a different blood type, antibodies in the recipient's plasma will bind to the antigens on the donor's blood cells, and an agglutination or cross-reaction will occur. This antigen-antibody complex will form a clump, or an agglutination. Anti-A or anti-B antibodies spontaneously develop during the first 6 months of life. Believe it or not, no exposure to foreign antigens is needed. But anti-Rh antibodies are different. Anti-Rh antibodies do not develop unless an individual is exposed to an Rh positive blood, any of those Rh antigens. Exposure can occur accidentally, during a transfusion, or during childbirth. That brings us to the last checkpoint of this lecture recording. See if you can test your knowledge and answer these questions before you move on to the next recording.